Okay, so we talked about DNA replication, which is how you copy DNA to make 2 copies of DNA so you can make 2 cells. An entirely different process the cells also do would be transcription and translation. These together are often called gene expression because it's how a gene gets expressed into a protein. Transcription is a process of using DNA as a template to make messenger RNA. Translation is a process of using messenger RNA to make protein. And just as a note, mRNA, a perfectly acceptable way to, because it takes way too long to write out messenger or say messenger. I'm just going to call it mRNA from now on. So RNA is a ribonucleic acid. It is a single-stranded nucleotide. It has a 5-carbon ribose sugar. It contains uracil instead of thymine. We discussed differences between DNA and RNA when we discussed our biomolecules. So I am expecting at this point that you have finished study of that chapter, so we're comfortable discussing RNA. There's 3 types of RNA that are used in our cells: rRNA, tRNA, and mRNA. Oh, that captioned awfully. Okay, rRNA is ribosomal RNA. It is what is going to combine with proteins to make ribosomes. Ribosomes are made of proteins and ribosomal RNA. tRNA, or transfer RNA, is what will transport amino acids. So the amino acids that are going to be used to make the protein, they're going to be brought to the ribosome by tRNA. And mRNA carries the genetic code. It has that message, the information from DNA, that it is going to bring that message to the ribosomes. While DNA is staying wrapped up in the nucleoid or in the nucleus, mRNA is going to come out to the cytoplasm, go interact with the ribosome, and be used to help make the protein. In transcription, DNA is transcribed to make messenger RNA. Messenger RNA is only going to be short-term. RNA is not terribly stable on its own, so messenger RNA is not going to stay around for a while. But it makes short-term copies of genes, so whatever that cell is needing to do at that moment, that RNA will provide a source for protein synthesis. Your gene on the DNA will start with a promoter sequence, and that promoter sequence will tell RNA polymerase this is where you need to bind. This is the start of the gene. So RNA polymerase will bind to the promoter sequence, and that will initiate transcription of the gene. Transcription will proceed in the 5' to 3' direction, adding nucleotides on the 3' end until RNA polymerase reaches the terminator sequence. And the terminator will terminate things. It stops transcription. So here is your strand of DNA, double-stranded double helix. And here in red is highlighted the promoter sequence. The promoter sequence is where RNA polymerase binds. And it separates these 2 strands of DNA from each other. And then RNA starts just base pairing, adding your sequence of A's, U's, G's, and C's on to the messenger RNA. And it just keeps on going until it hits the terminator sequence. So here you can see if the template strand of DNA has a T, the RNA is going to have an A. If it has a C, it'll put in a G. If it has a G, it'll put in a C. But if it has an A, RNA is now going to put in a U. So we'll have uracil instead. So when we are making our RNA, just remember that you can just copy the code on the template, or you can copy the code on the coding strand, or you can base pair with the template strand, but make sure you put in U's, uracils, instead of T's. Okay, then the terminator sequence is going to tell the RNA polymerase, all done, end of it. RNA polymerase can let go, and at the end you have a piece of messenger RNA. If you're a prokaryote, that's it, it's done. But if you're a eukaryote, we have a little bit of special stuff we're still going to do. In eukaryotic cells, we have what's called RNA processing. RNA processing is a really nice way that you can cut out different pieces of of that RNA transcript to end up with slightly different versions of a gene. So this one gene of DNA, you can have different pieces, be introns or exons, to make slightly different proteins. So our bodies are able to make multiple different proteins from one gene. RNA processing, though, only is performed in eukaryotes. Bacteria do not have the ability to do this, and it always happens in the nucleus. Within RNA processing, when this happens, we have exons and introns. Exons are the things that are expressed. These are the things that are going to exit the nucleus. These are the things that will make the protein. Introns are intervening or in the way. They stay in the nucleus. The introns are going to get chopped up. So here, your DNA, you have exons, introns, exons, introns. So in your RNA transcript, you have exons, introns, exons, introns. The introns are cut out, and the exons are put back together. So now you have your final messenger RNA that is ready. Now that it's been processed, it will exit to the cytoplasm and will be used for translation. There are SNRPs. SNRPs stand for small nuclear ribonucleoproteins. SNRPs are the proteins that are actually going to cut out the introns and put together the exons. And again, in a different situation, maybe this would be an exon and this would become an intron. You can make different versions of that protein depending on different sections if you're going to make it an exon or intron to have more variety of your proteins. Once your RNA is made, then it will go to the ribosome to be translated. Translation is going to decode the language of nucleic acids into the language of proteins. So messenger RNA is going to bring the code from the DNA. Transfer RNA or tRNA is going to bring the amino acids to the ribosome. And then what protein is going to be made is going to be dependent upon the genetic code. I have a genetic code worksheet practice put into D2L for you, and there is a separate video that is going to go through how to use the genetic code. So please make sure that you download that worksheet and you use that genetic code practice video to be able to go through that, because you will have questions on your exam that are going to say, here's a piece of DNA. What is RNA? Here's a piece of DNA. What will the protein be? You need to be able to do that kind of work. And that's what we talk about with that worksheet and that other video. Okay, back to your textbook chapter here. So in your messenger RNA, it's written in nucleotides, and our ribosomes are going to look at those nucleotides 3 at a time. So those 3 nucleotides at a time are called codons. That means there are 64 different codons. That is just 4 times 4 times 4. 64 codons. They're going to code for 20 amino acids. Of them, 61 are going to be sense codons, or code for an amino acid. So you can see here, these are sense codons. They code for phenylalanine. These are sense codons, code for leucine. These are sense codons, code for leucine. You may have noticed that genetic code is degenerate. Degeneracy in genetic code refers to its redundancy. So you could have this codon, this codon, this codon, this codon, this codon, or this codon. And all of those will give you leucine. Because we have 61 sense codons and only 20 amino acids, there's a lot of redundancy in here, or degeneracy within the genetic code. This is also kind of important because it gives your cells just a little bit of wiggle room. Just a little bit of mutation can happen where you still can end up with the same amino acid put in. So if this A turns into a G, you still can put in leucine. You haven't just lost the protein that you were making. Okay. The tRNA are going to be the actual translators, and the tRNA has what's called an anticodon. The anticodon is complementary to the codon. So the tRNA with its anticodon can actually bind to the mRNA with its codon, bind together, and be able to say which amino acid needs to be brought in. Translation always starts with this start codon, AUG. AUG always gives you methionine. So if I'm asking you, what is this messenger RNA going to be? It's always going to start with AUG. And if I'm asking you, what is this protein going to be? It always starts with methionine. If there is another answer, either you did something wrong or I wrote a bad question, I did something wrong. It always starts with AUG as the first codon, methionine as the first amino acid. Okay, and then translation is going to end at these nonsense codons, UAA, UAG, and UGA. So if you see UAA, UAG, and UGA, these are stop codons. When this codon gets into the ribosome, there is not a tRNA that will add an amino acid to it. Instead, that signals for the whole process, the ribosome, the messenger RNA, and the transfer RNA, it all just disassembles and translation stops at that point. Your protein has been fully made. Okay, so let's go through this process in pictures. Here is your messenger RNA. Here's its nice start codon, AUG. Here's your ribosomes. Remember, ribosomes have 2 subunits to it. And here's your tRNA. And this tRNA is going to be bound to different amino acids. So the whole thing joins together. And here's our start codon, AUG. So this tRNA has the anticodon, binds to it, brings in methionine. In this next section, we have the next codon. And the next amino acid can come in and bind to it because its anticodon is complementary to the codon. So it comes in and brings the next amino acid. We form a peptide bond between these 2 amino acids. And once that peptide bond is formed, the whole thing shifts down. So it shifts down. Now you have AUG in this exit site, the UA here in the middle, and a new codon ready to be read. So the next amino acid comes in. It can base pair its anticodon with the codon, brings in the next amino acid. It'll form a peptide bond. The old tRNA leaves from the exit site. The whole thing can shift down, bring in the next amino acid. Shift down, bring in the next amino acid. Shift down, bring in the next amino acid. On and on and on until you get to a stop codon. When you have a stop codon in the first site, it says, all done. And the whole thing disassembles. Your tRNA goes off. It can go get another amino acid to work again. Your mRNA goes off. It can be read by a ribosome again to make more proteins until it's degraded. Your ribosomes can come together and make more proteins. And this newly assembled amino acid can fold up and become a functional protein. So in eukaryotes and in prokaryotes, we already talked about a little bit difference between the two in terms of RNA processing. But there's also a little bit more that happens differently. So in eukaryotes, transcription is going to be in the nucleus. It involves DNA, so it has to stay in the nucleus. But translation is going to happen in the cytoplasm. So those two processes are separated in terms of where they happen, so they can't ever happen together. But in prokaryotes, both transcription and translation occur in the cytoplasm, and because they're occurring in the same space, they can occur at the same time. So here you have a cartoon. Here is DNA. You have RNA polymerase making these RNAs, and as soon as the RNA starts coming out, it's not even released yet. It's not even fully made yet. You've already got a ribosome jumping on it, starting to make a protein. So as transcription continues, your protein is just growing, making more proteins. So transcription and translation can happen at the same time. Here's a micrograph, an actual micrograph of here's the DNA, here's the RNA coming off, and you can see all these red ribosomes right there. Transcription and translation can be linked together. Okay, so now you can tell me what are the roles of the promoter, the terminator, and messenger RNA in transcription. You can tell me how does messenger RNA production in eukaryotes differ from the process of prokaryotes. You can tell me what it means to be degenerate. You can tell me what a start codon is, what a stop codon is. You can tell me the roles of those different types of RNA. And next you're going to go watch the video about how to read the genetic code, how to create RNA and proteins. So when I ask you that question, you'll be able to answer that. And then I'll see you back. We'll talk about mutations. Okay, bye.